This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas.
Right now, literally right beneath your feet, there is just this raging, completely invisible war going on.
A massive microscopic war.
Yeah, exactly. It's a conflict of staggering proportions.
Yeah.
And it's happening constantly in every single city on Earth.
Right under our streets.
Right. You've got trillions of biological entities that are just, hunting, evading, and destroying one another.
It's a very violent ecosystem down there.
It really is. And I think the most fascinating part about this war is that it's being fought in a language that until very recently, scientists couldn't even read.
Not even a little bit. It was total static.
Which is wild. Because if you were to say, scoop up a cup of untreated city wastewater, right, just raw sewage.
Just a standard cup from a treatment plant, yeah.
Yeah, and if you sequence all the genetic material inside that cup and then ask the world's most powerful supercomputers to identify what you just found, well, they would give you a pretty startling result.
A very frustrating result if you're a bioinformatician.
Yeah.
Seriously. Because on average, about 56% of the genetic material in that single cup is just unclassifiable dark matter.
More than half. Just totally unknown.
More than half of what is flowing beneath our streets every time you flush the toilet is a complete biological mystery.
Which is, it's actually a deeply unsettling statistic when you think about it.
Oh, absolutely.
Because public health officials are increasingly relying on that exact same water to tell us if, say, the next global pandemic is brewing in our neighborhoods.
Right, they want to use it as an alarm system.
Exactly. We are trying to use this massive, incredibly complex biological ecosystem as an early warning system. But we only actually understand a fraction of what lives inside it.
And that perfectly sets up our mission for this deep dive today. We are going to illuminate that dark matter.
We are diving right into the deep end of the sewers.
We really are. We've got a fascinating stack of sources in front of us, but our anchor today is this groundbreaking scientific paper. It's titled A Genome-Resolved View of the Wastewater RNA Virome. It's a phenomenal piece of work.
It is. And the researchers behind this, scientists like Rose Cantor, Bigan Shakia, Mark Johnson, and this entire network known as the CRISPR Consortium, they didn't just write some dry academic text.
No, not at all. It reads more like a detective story.
It really does. They documented this monumental, almost unbelievably complex journey. They literally set out to catalog the invisible ocean of viruses flowing through our municipal pipes.
It was a massive undertaking.
So we're going to explore the grueling journey they took to map this unknown universe. We'll look at the dead ends they hit and how their ultimate success is currently being deployed to protect you from the next catastrophic disease.
And we also really have to grapple with the ethical side of this.
Oh, for sure. The ethical tightrope we walk when our technology allows us to look this closely at the collective biological output of society.
Because the implications for privacy are huge.
But before we get to the supercomputers and the ethics, let's step back. To appreciate the sheer scale of what this CRISPR Consortium achieved with their new database, we kind of have to look backward first, right?
We do, because the whole concept of looking at human waste to track human health, which we officially call wastewater-based epidemiology, or WBE, well, it sounds so futuristic.
Yeah, it sounds like cyberpunk. Like the city grid is constantly monitoring your internal biology.
Exactly. But the foundational logic of WBE is actually quite old.
Right. I mean, we all kind of know the famous story of Dr. John Snow, right, back in 1854.
The Broad Street Pump.
Yeah.
Classic epidemiology.
Right. He was mapping out that horrific cholera outbreak in London's Soho district, and he famously figured out that all the deaths were clustered around this one specific water pump on Broad Street.
Which was revolutionary because at the time, everyone thought disease spread through miasma.
Miasma, yeah, like bad air.
Right. But Snow realized the water was contaminated with waste, so he roofed the pump handle and boom, stopped the outbreak.
Epidemiology 101. But when I was reading through our sources, I was really surprised to learn about a much more direct ancestor to modern wastewater sequencing. And it didn't happen in the 1800s.
No, it really picked up in the mid-20th century.
Right, the 1940s.
The 1940s polio epidemics. That era is truly the birth of virological wastewater surveillance.
Okay, so how did that work? They definitely didn't have high-speed genetic sequencers back then.
Oh, definitely not. But in the late 1930s, researchers made this crucial discovery. They actually successfully isolated the poliovirus from urban sewage.
Wow, just pulling it straight out of the muck.
Exactly. And polio was this unique public health terror at the time, mainly because of how it presented clinically.
What do you mean?
Well, for every single patient who developed that tragic, highly visible paralysis, there were potentially hundreds of other people in the community who were infected and actively shedding the virus in their waste.
But they weren't paralyzed.
Right. They were showing absolutely no symptoms, or maybe just had a mild fever.
So if you're a doctor back in the 1940s trying to track a polio outbreak, and you're only looking at the paralyzed patients in the hospital, you're really just looking at the very tip of a massive hidden iceberg.
You're completely blind to where the virus actually is in the community. Clinical surveillance completely fails in that scenario.
So what do?
Public health officials turned to the sewers. They developed this incredibly simple but effective tool called a Moore swab.
A Moore swab. Okay, paint a picture for me. What does that look like?
It is essentially a specialized, tightly bound cotton pad tied to a very long string.
Seriously, just cotton on a string.
Literally, they would just drop these swabs down into the sewer grates of specific neighborhoods, leave them there for a few days to absorb the flow of whatever went by, and then haul them back up.
Oh, man. I can only imagine what the laboratory process for that was like. That sounds so incredibly gross.
It was incredibly laborious and, yes, very gross. They had to take these heavily contaminated, soaking wet swabs back to the lab.
And then what? Just squeeze them out.
Sort of. They'd extract the liquid, but then they had to treat it with massive amounts of antibiotics.
Oh, to kill off all the regular bacteria in the sewage.
Exactly. Because they only wanted the viruses. Once they killed the bacteria, they would introduce that extracted material into tissue cultures, or sometimes animal models.
Right, and then just wait and see if the virus is there.
Basically. They had to wait to see if the cells exhibited cytopathic effects, which is just a fancy way of saying they watched to see if the cells died. If the cells died, it confirmed the poliovirus was present in that neighborhood.
How long did that whole process take?
Weeks. It was very slow. But despite that slow turnaround, it proved this revolutionary concept. It proved you could monitor the unseen spread of a virus across an entire population without needing a single person to go visit a doctor.
That's incredible.
It really is. And tracking polio in wastewater is still a highly sensitive, highly critical practice today, especially in parts of the world where the virus is near eradication.
So the scientific community has known this worked for a long time. The proof of concept existed for decades. But reading through the historical context in our sources, WBE still seems like a pretty niche subfield.
Yeah, it was mostly environmental scientists and academics doing it.
Right, it wasn't exactly front-page news. But then, obviously, the year 2020 arrived.
Like everything changed.
The COVID-19 pandemic completely shattered the status quo for this field.
It forced a massive global paradigm shift. I mean, within a matter of weeks, wastewater testing went from this quiet academic pursuit to arguably the most vital public health intelligence tool on the planet.
And why was it adopted so fast? I mean, beyond the obvious crisis of the pandemic.
Because it completely bypassed the inherent massive failures of clinical testing during a crisis.
Oh, right, because early in the pandemic, let's be honest, getting a clinical test was like finding a golden ticket. It was impossible.
It was a nightmare. Let's break down those failures because it really highlights the genius of wastewater tracking. First, to get a clinical test, you had to physically feel sick enough to even want one.
And a lot of people were asymptomatic, so they didn't even try.
Exactly. But let's say you felt sick. Then you had to have the financial means, the reliable transportation, and the time off work to actually go to a testing site.
Assuming there was even a site open near you?
Right. You had to hope they had tests available. Then you had to endure the incredibly uncomfortable swab by a healthcare worker.
The brain tickler.
Yes, the deep nasal swab. Then that swab had to be packaged, transported safely to a lab, processed by backlogged technicians, and eventually reported back to your local health department.
Every single step in that chain is a huge bottleneck.
Massive bottlenecks. And more importantly, every single one of those steps introduces profound behavioral and socioeconomic biases into the data.
What do you mean by biases?
Well, think about it. If you lack health insurance, or if you're an hourly worker who simply cannot afford to miss a shift, you probably aren't going to wait in line for 6 hours to get tested.
Right, because if it's positive, you have to isolate and lose pay.
Exactly. So those populations become completely invisible data points to the health department. Your illness goes unrecorded, and the public health officials think the community is safer than it actually is.
But the wastewater grid, it doesn't care about your insurance status.
Not at all.
It doesn't care if you have time off work. It captures absolutely everyone who is connected to the plumbing system.
Everyone. Furthermore, it captures the biological output of all those asymptomatic individuals we mentioned and the pre-symptomatic individuals who are actively shedding the virus but don't even know they're sick yet.
So they're flushing evidence before they even have a cough.
Precisely. And within days of the global pandemic declaration, scientists confirmed they could easily detect fragmented SARS-CoV-2 RNA in community wastewater.
It was a total game changer.
Suddenly, health departments had this completely unbiased, real-time picture of community spread. They were seeing spikes in the wastewater viral load days, and sometimes even weeks, before those cases started showing up at the hospital emergency rooms.
Okay, let me try out an analogy here, just to make sure I am fully grasping the mathematical power of this aggregate testing.
Let's hear it.
So, if clinical testing is kind of like a botanist trying to assess the health of this massive million-acre forest, right? And they're just walking around on the ground, examining individual leaves on individual trees, hoping to stumble upon the diseased ones.
A very slow process.
Is wastewater testing like taking a high-resolution satellite photograph of the entire forest canopy all at once?
The satellite analogy captures the scale perfectly, but we need to push it just a little bit further to really understand the mechanics of the chemistry involved.
Okay, how so?
Well, a satellite photo might still let you zoom in and look at an individual tree if the resolution is good enough. Wastewater doesn't do that at all. It destroys individuality.
Oh, because it all mixes together in the sewer?
Exactly. So a better way to look at it is, imagine taking all the leaves from that entire million-acre forest, tossing them into a massive industrial blender, and turning it into a green smoothie.
Okay, slightly gross, but I'm with you.
And then you take just a single drop of that blended mixture to analyze its chemical makeup in the lab.
Wow.
Yeah, it completely destroys the individual identity of any specific tree. You can't say, this oak tree is sick, but that single drop gives you the exact aggregate health status of the entire ecosystem simultaneously.
It provides spatial and temporal information on the entire contributing population from just one composite sample.
Exactly. That is the true power of WBE.
That makes a lot of sense. The power is entirely in the aggregate. But as we transition into the primary research paper driving our deep dive today, the Casper Consortium's paper on the RNA virome, we quickly learn that analyzing that blended drop of forest is astronomically difficult.
It is a bioinformatic nightmare.
Right. Because the researchers looking at the wastewater now, they aren't just trying to measure COVID-19 anymore. We know how to do that. They are hunting for something infinitely more elusive.
They are hunting for disease X.
Which I have to say sounds like a comic book villain.
It does sound theatrical, but it's a very real, very serious public health term.
Right. Who coined that?
It's the placeholder designation adopted by the World Health Organization.
Okay. And what does it mean exactly?
It represents the terrifying reality that a serious, catastrophic international epidemic could be caused by a pathogen that is currently completely unknown to human science.
So it's the nameless virus that will cause the next pandemic.
Exactly. It might be a novel coronavirus we've never seen, or maybe a highly pathogenic strain of avian influenza that suddenly mutates to transmit easily between humans. It could be a new hemorrhagic fever, or a totally uncharacterized zoonotic virus that jumps from an animal reservoir into a human host.
So the scientists in the Casper Consortium, Cantor, Shakya, Johnson, and all their colleagues across the country, they look at the wild success of wastewater tracking during COVID, and they say, hey, let's use this incredible early warning system to catch disease X before it spreads.
That was the dream, yes.
But almost immediately, they run headfirst into a brick wall, a biological roadblock that stems from what wastewater actually is.
Right, it's an assumption problem.
Because when the average person thinks of a sewer, they think of human waste, obviously. Therefore, it's completely logical to assume that the water down there is just absolutely teeming with human-infecting viruses.
It's a very logical assumption, but it turns out it is entirely incorrect.
Wait, really?
Yes. In the vast, complex soup of municipal wastewater, vertebrate-infecting RNA viruses.
Meaning the ones that infect humans and animals.
Right. The specific types of viruses that actually cause diseases in us, like influenza, SARS-CoV-2, or norovirus, They are incredibly rare in the sewers.
Rare. I thought they would be everywhere.
No, they represent a minuscule fraction of the overall genetic material floating in that water.
Okay, but if the viruses that make us sick are just a tiny drop in the bucket, what is taking up all the rest of the room? What is the dominant life form down there?
The wastewater virome is overwhelmingly dominated by two main things. The first, and by far the largest, are bacteriophages.
Bacteriophages. Okay, break that down for me.
These are viruses that exclusively hunt, infect, and replicate inside bacteria. They don't touch human cells at all.
Ah, okay. And because human waste is full of bacteria.
Exactly. Human waste is essentially just a massive vehicle for trillions of gut bacteria. When those bacteria enter the sewer system, they encounter this environment that is incredibly rich in organic matter.
A buffet for bacteria.
Right. So they can sometimes continue to multiply in the pipes, and consequently, the viruses that prey on those specific bacteria, their population just explodes.
So the sewers are essentially this massive microscopic battleground between bacteria and phages.
It is a nonstop microscopic war zone. That's the first dominant group. But the second dominant group of viruses in the water is really surprising.
What is it?
Plant viruses.
Wait, plant viruses surviving in human sewage.
Massive amounts of them.
How does a virus that infects, like a tomato plant, end up dominating a municipal water treatment facility?
Through the human diet. Yeah. When you sit down and eat a salad or some salsa or really anything containing raw plant matter, you are ingesting millions and millions of plant viruses.
I had never even thought about that.
Most people haven't. And here's where the evolutionary biology becomes truly fascinating. The protein shells, which we call capsids, that encapsulate the genetic material of many plant viruses are extraordinarily robust.
I guess they have to be to survive out in a field.
Yeah,
Right.
exactly. They have to survive harsh sun, soil, water, agricultural environments. But that same evolutionary toughness allows them to survive the highly acidic environment of the human stomach.
Oh, wow. So they just pass right through us.
They resist the corrosive stomach acid. They resist the digestive enzymes in our intestines. They pass entirely through our gastrointestinal tract completely unharmed.
And then into the toilet.
Right. And from there, they survive the harsh chemical and physical conditions of miles of sewer pipes. They don't infect human cells. They don't make us sick in the slightest, but they leave an absolutely massive genetic footprint in our waste.
Okay, so let's reset the board here. These researchers want to find disease X. They are looking for a microscopic needle, this incredibly rare, currently unknown, potentially pandemic-causing human virus.
Yes.
And that needle is buried inside a colossal haystack made almost entirely of bacteria-infecting phages and indestructible plant viruses from our lunch.
That is the exact problem.
So how did scientists attempt to find these needles before the CRISPR Consortium published this paper? What was the old way of doing things?
Well, the standard technology, which is still widely and very effectively used for pathogens we already know about, relies on targeted methods, specifically techniques like probe capture, hybridization, or targeted amplicon sequencing.
Okay, I'm definitely going to need a translation for probe capture or hybridization. Let's stick with the haystack analogy if we can. Is probe capture like using a highly customized magnet to pull the needle out?
A magnet isn't quite the right way to visualize it, mostly because a magnet pulls all ferrous metal indiscriminately. Probe capture is much, much more precise than that.
Okay, what's a better analogy?
It is much more akin to using a molecular barcode scanner.
A barcode scanner.
Right. In the lab, scientists synthesize these tiny complementary fragments of genetic code, these are the probes, and they design these probes to perfectly match the specific genetic sequence of the virus they want to find.
Oh, I see.
So these probes act exactly like a barcode scanner at a grocery store checkout. You scan a massive pile of mixed groceries, that's your haystack of wastewater, and the scanner only beeps when it registers the exact pre-programmed barcode for a Granny Smith apple.
And it just totally ignores the oranges, the cereal, and the milk.
Exactly. So in the lab, you design specific probes for SARS-CoV-2, you introduce them into your blended wastewater sample, and those probes bind to or capture only the COVID RNA.
Allowing you to pull it out and sequence it, while completely ignoring the billions of phages and plant viruses.
You've got it. It's incredibly efficient.
That sounds amazing if you want to track a known virus like COVID. But I can immediately see the critical flaw if you are out there hunting for disease X.
The flaw is glaring.
Right. Because a barcode scanner can only beep if the item is already registered in the grocery store's database.
Precisely.
If disease X is a completely novel virus, a virus that has never infected humans before, Science doesn't have its barcode yet. We don't know its genetic sequence, so we can't possibly design a probe to capture it.
That is exactly the problem. Targeted methods are inherently backward-looking. They can only monitor what we already know to fear.
They will completely, utterly miss a novel emerging pandemic threat.
They'll miss it entirely because the specific probes will just wash right past the unknown RNA. To find disease X, you have to throw away the barcode scanner. You cannot use targeted methods at all.
So what do?
You have to look at absolutely everything in the sample. You have to use a technique called untargeted ultra-deep sequencing.
Untargeted sequencing, meaning you don't use a probe, you just dump the entire cup of wastewater into a high-tech machine and tell it to read every single piece of genetic material it can find.
Yes. Regardless of whether it's a human virus, a plant virus, or a phage, you sequence it all. It is completely agnostic.
Which sounds like the ultimate solution for an early warning surveillance system.
It sounds perfect in theory, but when the scientific community first attempted this untargeted approach with wastewater, they ran headfirst into a massive computational barrier. They hit what bioinformaticians call the read-based wall.
The read-based wall. Okay, let's explore this, because reading the paper, this is where the sheer scale of the data just starts to become completely mind-boggling. First off, what exactly is a read?
Okay. When you put genetic material into a modern next-generation sequencing machine like an Illumina sequencer, which is kind of the industry standard right now, the machine does not spit out one long, perfect, continuous string of code representing a full viral genome.
It doesn't just print out the whole virus from start to finish.
No, the chemistry of the sequencing process physically requires the DNA or RNA to be broken up into tiny pieces first. So the machine reads these tiny pieces, producing millions of short, fragmented strings of genetic code.
How short are we talking?
Typically only about 150 base pairs long, very small. Each of these short fragments is called a read.
So you start with complete microscopic viral genomes floating in the water, but to analyze them, the machine forces you to shatter them into millions of tiny puzzle pieces.
Yes, that's a great way to think about it. And historically, the standard bioinformatics pipeline to make sense of these millions of scattered puzzle pieces was to use a reference database.
How does that work?
You take each 150 base pair read and you run it through a massive computer database of every known viral genome ever published by science. You ask the computer, does this short sequence match any part of the Ebola genome? Does it match the measles genome? Does it match the tomato mosaic virus?
So it's like having a single sentence torn out of a book and using a massive library index to figure out which book that one sentence came from.
Exactly. But this is where the dark matter we talked about earlier rears its ugly head. When the researchers ran their untargeted wastewater reads through these global reference databases, the computers essentially just shrugged.
They couldn't find a match.
On average, 56% of all the reads in their data were totally, completely unclassifiable.
More than half of the data returned zero matches.
Zero.
To visualize this, imagine you are handed a sprawling ancient library containing tens of thousands of different books.
All right.
But before you get them, the entire library is put through an industrial paper shredder. You are handed billions of tiny, shredded strips of paper.
A nightmare scenario.
Right. And you try to reconstruct the books by matching the text on the shreds to known texts. But you discover that 56% of the shreds are written in an alien language that no human being has ever seen before.
That analogy perfectly captures the despair of the bioinformatician, and think about the real-world public health implications of that alien language.
It's terrifying.
It is, because if 56% of your shreds are entirely unknown, how can you possibly triage a threat? Let's say an anomaly pops up in your data, some weird spike. How do you know if that strange new sequence belongs to a harmless, undiscovered virus that only infects a rare type of soil bacteria, or if it belongs to a highly lethal, newly mutated respiratory virus that is currently incubating in your city?
You can't. You'd have no idea. The read-based method basically cannot separate the benign background noise of the dark matter from the catastrophic signal of disease X.
It's just a wall of impenetrable static.
Which is the exact problem the Casper Consortium set out to solve with this paper.
Exactly. They realized a fundamental truth of surveillance. To find the dangerous unknown, you first have to meticulously map the harmless unknown.
You have to translate the alien language.
You have to illuminate the dark matter.
And that monumental realization brings us to the core of our primary paper today. The grueling, resource-intensive, almost unimaginable journey to build the WVDB, the wastewater virome database. They just decided to map the dark matter themselves.
The ambition of this project, I really can't overstate it. The CASPR researchers didn't just analyze a few vials of water from their local university campus, they orchestrated a massive nationwide collection effort.
Right, the scale is wild.
They performed untargeted ultra-deep sequencing on 321 separate untreated wastewater samples.
And these weren't all from one place, right?
No, these samples were meticulously collected from 11 different locations across 6 different U.S. cities, spanning a period between early 2023 and early 2025.
So they were trying to get a really broad picture.
Very broad. They sampled major metropolitan hubs like Chicago and Los Angeles to capture highly dense, diverse urban populations. But they also ran a 1-year weekly time series sampling protocol in Columbia, Missouri,
Why Missouri?
to track how the virome changes over the seasons in a more mid-sized city. And my favorite part, they even heavily sampled an international airport in Chicago.
Oh, to capture the genetic signatures of global transit, people flying in from all over the world.
Exactly, the ultimate mixing pot.
You mentioned ultra-deep sequencing a second ago, and the numbers in the paper for this are just staggering. They weren't just taking a quick snapshot of the water. The paper states they were aiming for a median depth of 1.6 billion reads per sample.
1.6 billion individual shattered fragments of genetic code, pulled from a single sample, and they repeated that across 321 samples.
So we are talking about hundreds of billions of individual data points.
Hundreds of billions. It is an ocean of genetic information so incredibly vast that it pushes the limits of modern computational infrastructure to even physically store it on a hard drive, let alone analyze it.
So they have this mountainous pile of shredded paper, hundreds of billions of shreds, and we already established they cannot just compare them to a reference dictionary because the alien words aren't in the dictionary.
Right.
So how do you possibly begin to read a shredded book when you don't know the language?
You have to abandon the reference dictionary entirely, you throw it out, and instead you turn to a fiercely complex computational process known as de novo assembly.
De novo, which is Latin for from the beginning or anew.
Exactly, or, to put it simply, from scratch. Instead of taking a shredded read and looking for a match in an external database, you feed all 1.6 billion shreds from a single sample into a supercomputer.
Okay.
And you ask the algorithms to look strictly for internal overlaps.
Wait, how does a computer actually do that? Because finding overlaps in a billion tiny pieces sounds like it would take 1,000 years to process.
It requires immense processing power and truly brilliant mathematics. The algorithms use these complex data structures called de Bruijn graphs.
De Bruijn graphs.
Yes. Imagine you take a single shred that is 150 letters long. The computer scans the entire data set looking for another shred where, say, the last 149 letters perfectly match the first 149 letters of a different shred.
Okay, looking for exact text alignments.
Right. If it finds that perfect staggered overlap, it stitches them together to make a slightly longer sequence. Then it looks for another shred that overlaps the end of that new sequence.
And it just keeps going.
Slowly, painstakingly, using sheer computational brute force, the algorithms stitch the short reads into longer and longer continuous sequences. These long sequences are called contigs.
Contigs? So they are essentially reconstructing the pages and eventually whole chapters of the books from scratch without ever having seen the cover.
That's exactly what they're doing.
That is genuinely mind-blowing to me. It's like solving a billion-piece jigsaw puzzle where the pieces are all mixed together from 1,000 different puzzles and the pieces are microscopic and you don't have the picture on the box for a single one of them.
It is an absolute miraculous feat of bioinformatics, but as you can imagine, it is fraught with peril.
I bet.
RNA viruses are notoriously difficult to assemble this way. For one, their genomes are relatively small compared to bacteria, which just gives you fewer overlapping pieces to work with.
Less data to overlap.
Right. Furthermore, RNA is a very fragile molecule. It degrades rapidly in harsh environments.
And a sewer is definitely a harsh environment.
Exactly. By the time that RNA travels through miles of sewer pipes, exposed to wild changes in temperature, chemical cleaners people pour down their sinks, UV light if any channels are open, and enzymes called nucleases that actively chew up RNA.
The strands are probably heavily damaged.
They are beaten up. Plus, the sheer diversity of viruses in the water means many of those tiny puzzle pieces look incredibly similar to one another.
Which introduces the potential for catastrophic errors, I would assume.
Yeah.
When you are blindly stitching things together based purely on overlapping letters, you risk making mistakes.
Huge mistakes. And the paper actually dedicates a significant amount of ink to the rigorous quality control they had to implement to avoid publishing what they call chimeric sequences.
Chimeric sequences. Let's dig into that. What exactly makes a sequence a chimera in this genomic context?
Well, think about classical mythology. A chimera is a monstrous creature made of incongruous parts, right? Like the head of a lion, the body of a goat, the tail of a serpent. In genomics, a chimeric sequence is basically an accidental Frankenstein virus created by a computational glitch.
How does that happen?
It happens when two completely different viruses happen to share a very short sequence of identical genetic code by sheer coincidence. The de novo assembly algorithm sees that identical overlap and it gets confused. It mistakenly bridges the gap, stitching the left half of, say, a harmless plant virus to the right half of a dangerous human virus.
Oh, wow. So it creates a genetic sequence for a virus that doesn't actually exist in nature.
Precisely. And in the high-stakes hunt for disease X, publishing a chimeric sequence in a global database is incredibly dangerous.
I can see why. Imagine the global panic if a database falsely reports the discovery of a highly transmissible airborne respiratory virus that somehow fused with the hemorrhagic lethality of Ebola.
And then it turns out to be nothing more than a math error in an overlap graph. It would be a disaster.
The researchers knew this new database had to be absolutely bulletproof to be trusted by the global health community. So, to purge the chimeras, they instituted a devastatingly strict rule for inclusion.
It was a brutal standard, truly. For a newly assembled viral genome to be certified and officially included in the final wastewater virome database, it had to assemble completely as a single contiguous sequence independently at least twice across their different samples.
Wait, so if a totally unique, fascinating virus assembled perfectly from the shreds in a sample from Chicago, but they never saw it assemble completely again in any other sample across the country.
It was thrown out, discarded entirely.
Wow.
Because of this incredibly strict multi-sample validation rule, the team had to throw away over 21,343 unique, fully assembled sequences.
They threw away over half of their potential discoveries. That has to be agonizing for scientists who spent months crunching that data on a supercomputer.
It requires immense scientific discipline. They actively sacrificed volume for absolute certainty, and our sources detail other painful sacrifices they had to make because of this methodology.
Like what?
Well, because they strictly required single complete contigs, meaning one long unbroken string of RNA to prove it was a whole virus, they systematically missed segmented viruses.
Segmented viruses, like influenza.
Yes, the influenza virus is a classic example of this. It doesn't keep its genome on one single continuous string of RNA. It keeps its genetic code distributed across 8 separate, distinct segments.
Almost like separate mini chromosomes.
Exactly. So because the assembly algorithm was explicitly looking for one long continuous string, the separate segments of the flu virus couldn't be easily reconstructed into a single genome, so they were largely missed by this specific database build.
That's a pretty big blind spot.
It is, and they also missed any viruses that were present in the wastewater at such low abundances that there simply weren't enough overlapping reads to physically reconstruct the full genome.
Right, if you don't have enough puzzle pieces, you can't build a picture.
Exactly, but honestly, it is a testament to how meticulous their science was. They accepted the limitations of their method to ensure the absolute integrity of their results.
Quality over quantity.
Exactly. And despite all those dead ends, despite the heartbreaking deletion of 21,000 sequences, the project was a monumental triumph.
They successfully finalized the first version of the WVDB.
They did. They confirmed 21,015 unique, high-quality, near-complete viral genomes.
That number represents a staggering leap forward in humanity's understanding of the microbial world. Because to put the novelty of their achievement in perspective, out of those 21,015 genomes they painstakingly pulled from the dark matter, fewer than 4,000 of them had any matches, even at the broad genus level, in any previously published global virus database.
Let that sink in.
Let me pause on that. You were saying that roughly 17,000 of the viral genomes they reconstructed from the sewage had literally never been seen, cataloged, or identified by human science before this paper was published.
That is correct. They essentially discovered, mapped, and published the genetic blueprints for 17,000 completely novel biological entities.
That is wild.
They pulled an entire undiscovered universe out of the sewers and into the light.
This is where the science gets truly fascinating to me. What exactly did they find in the dark? If we look at this newly illuminated cast of characters, what is actually making up this unseen universe beneath our cities?
Well, the data spectacularly confirmed what we suspected earlier about the extreme rarity of vertebrate viruses.
Right.
Out of the 21,015 viral genomes finalized in the database, a massive 79% were bacteriophages.
79%?
Yes. Specifically, they belong to a class of positive-sense, single-stranded RNA bacteriophages called liververicutes.
Nearly 8 out of every 10 viruses they cataloged is a phage that exists solely to hunt bacteria. The paper notes they fell largely within these taxonomic orders called Norsavirales and Tinlavirales.
Right.
It's just wild to visualize the sewers not as, a static flow of waste, but as this hyperactive, dynamic predator-prey ecosystem where trillions of viruses are constantly attacking the gut bacteria we flush away.
It is a vibrant, chaotic, microscopic jungle down there. But honestly, the sheer volume of phages was somewhat expected by the scientific community. The other major dominant group was the truly surprising one.
Right, the dietary culprits. I found this section of the data absolutely captivating when the CRISPR team looked across all 11 of their sampling sites.
Trying to find the most ubiquitous, highly abundant viruses that showed up everywhere.
Yeah, the ones that were everywhere, from Chicago to Missouri to Los Angeles. The winners were not human pathogens, they were plant viruses.
Specifically, they found massive abundances of species within the tabemovirus genus.
The tabemovirus.
Yeah. The paper explicitly catalogs incredibly high read counts for things like the pepper mottle virus, the tomato mosaic virus, and the cucumber green mottle mosaic virus.
Okay, so to trace the journey of that RNA, someone in Chicago sits down and eats a salad containing tomatoes and bell peppers.
Yep.
The viruses infecting the cells of those specific plants survive being chewed by human teeth. They survive the highly corrosive hydrochloric acid in the human stomach. They withstand the absolute barrage of digestive enzymes in the small intestine. They make the whole journey through the municipal plumbing, and they emerge at the other end as the loudest, most abundant genetic signal in the entire wastewater treatment plant.
Biology is remarkably resilient, yes. The evolutionary architecture of the tabemovirus capsid, that tough outer protein shell we mentioned, is incredibly dense and stable.
It's basically biological armor.
Exactly. It evolved to protect the fragile viral RNA in harsh agricultural environments, like in the soil or sitting in irrigation water. That exact same armor allows it to pass entirely through our bodies completely undegraded.
So they don't infect us at all.
They do not interact with human cells, but they leave a massive, unavoidable genetic trace in our waste.
It really makes you contemplate the hidden biological complexity of your lunch.
It does. But of course, mixed in with the millions of phages and the indestructible pepper viruses, the researchers did actually locate the human pathogens they were searching for.
They did. By matching their assembled genomes against known clinical databases, they successfully recovered the genomes for classic human enteric viruses.
The familiar pathogens that cause gastrointestinal distress and stomach flu.
Right, the stuff that makes us miserable. The database includes robust representations of norovirus, sapovirus, various enteroviruses, astrovirus, and cobuvirus.
All present and accounted for.
Okay, so they successfully cataloged the phages, the plant viruses, and the stomach bugs. But let's circle back to the grand public health mission we started with.
The Hunter disease X.
Exactly. Why does a public health official, someone whose literal job is to prevent the next respiratory pandemic, care deeply about a database filled with 16,000 harmless bacteria-infecting phages and the tomato mosaic virus?
Because of the noise-canceling revelation.
This is my favorite part.
This is the true world-changing genius of the wastewater virome database. The database itself isn't meant to be a list of threats. It is the ultimate calibration tool.
If I could try another analogy here to help explain this concept. Think about a pair of high-end active noise-canceling headphones.
Okay, I like where this is going.
If you are sitting on an airplane, those headphones don't just put a physical block of foam over your ear to muffle the roar of the jet engines. That's passive isolation.
Right, they actively fight the sound.
They use tiny microphones on the outside to actively listen to the specific frequencies of the engine noise. Then, a computer chip inside the headphones generates a sound wave that is the exact inverse, the exact opposite of that engine noise.
It flips the wave upside down.
Exactly.
Yep.
It plays that inverse wave into your ear, and the two sound waves physically cancel each other out in the air. The deafening roar of the jet vanishes, and suddenly you can clearly hear the delicate, quiet notes of the acoustic guitar track you are trying to listen to.
That is precisely what the WVDB allows bioinformaticians to do with genetic data. It is literally a digital noise-canceling filter for wastewater.
So before this database,
Before this paper was published, if a scientist ran untargeted sequencing on a wastewater sample, they were hit with a deafening roar of 56% unknown static.
that read base wall we talked about.
Yes. If the tiny, delicate genetic signature of a novel disease X was hiding in that sample, it was entirely invisible. It was completely drowned out by the roar of 17,000 unknown phages and plant viruses.
But now armed with this database.
Now, scientists possess the exact genetic blueprints for that background noise. When the next wastewater sample comes in and the sequencer produces 1 billion reads, the algorithms first run those reads against the WVDB.
They look for the noise.
Exactly. The computer identifies every single read that belongs to a newly cataloged phage or a pepper virus, and it digitally subtracts them. It filters them out of the data set entirely. They press the noise cancel button on the dark matter.
And when the noise clears?
When the background noise is digitally silenced, anything truly novel left behind, anything that doesn't match the WVDB and doesn't match known human reference databases, stands out glaringly against a blank digital background. If an unknown, heavily mutated respiratory virus begins circulating in a community, its genetic fragments will no longer be lost in a sea of unclassifiable static. It will trigger an immediate, undeniable anomaly in the data. The WVDB is the prerequisite tool required to make untargeted early warning systems actually function in the real world.
So we have this incredible database sitting on a supercomputer. We have the digital noise-canceling filter. But how does this brilliant science actually translate into saving a patient's life?
That is the ultimate question, isn't it?
Right, because a database doesn't administer a vaccine or free up a bed in a hospital ICU. How do we take this massive stream of complex genetic data and actually deploy it into public policy?
To understand the deployment, we have to look at the supporting sources in our stack. These detail how this theory is being applied on the ground, in the real world, right now. We look to the pioneering work being done by the Texas Epidemic Public Health Institute.
TI?
Yes, TTI. And specifically, their establishment of the Texas Wastewater Environmental Biomonitoring Network, or TexWWeb.
TexWeb is a fascinating case study because it represents this messy, real-world collision of high-level academia and local civic administration.
It really does. It's a massive collaborative network spanning 15 cities and 38 monitoring sites across the entire state of Texas.
That's a huge operation.
It involves bioengineers pulling the samples from the grates, statisticians crunching the data on servers, local health department directors trying to interpret what it means, and then medical clinicians actually treating the patients in the hospitals.
And the primary hurdle they faced, which our sources highlight, was data paralysis.
Yes. Information overload.
You simply cannot hand a city mayor a spreadsheet containing the fluctuating read counts of 20,000 different viruses and expect them to craft a coherent public policy.
Raw data without context is paralyzing. It leads to total inaction. So to solve this translation problem, the TTI Wastewater Consortium's Action Plan Work Group realized they had to design a triage system.
A way to rank the threats.
Exactly. They developed a matrix that categorizes the overwhelming stream of viral data into 5 distinct, actionable tiers of threat. This categorization is the vital bridge between the laboratory and the local clinic.
Okay, let's explore how this triage matrix works. Because if you are running TexWWeb, how do you avoid calling the governor in a total panic every time someone gets the sniffles? Let's start with Category 1.
Category 1 handles the endemic or seasonal viruses.
Meaning the ones that are always around?
These are the pathogens we fully expect to see. Influenza, RSV, and now SARS-CoV-2. They circulate in very predictable seasonal waves. Public health officials monitor these signals as lead indicators.
So it's not about stopping them completely.
No, the goal isn't to sound a catastrophic alarm. It is purely resource management. If the wastewater signal for RSV begins a steep upward curve in early October, hospital administrators know they have roughly 10 to 14 days to proactively increase pediatric bed capacity.
And pharmacies know to order larger stocks of antiviral medications and inhalers before the rush hits.
Exactly, it's logistics. Then we have Category 2, which are the sporadic viruses.
Sporadic, meaning unpredictable.
Yes. These are primarily the gastrointestinal threats, norovirus, adenoviruses, rotavirus. They cause very significant outbreaks, but their timing is irregular. They don't follow a strict season.
Again, you don't shut down a major city for a norovirus outbreak.
Right. But if the wastewater signal hits a predetermined high threshold in a specific region, the health department can execute targeted interventions.
Like what?
They might send out official advisories to local nursing homes, daycares, and schools in that specific sewer shed, urging them to implement stringent hand washing and surface sanitization protocols.
Ah, trying to blunt the impending outbreak before it peaks and hits the vulnerable population.
Yeah, precisely. Now, the temperature rises significantly at Category 3. These are the vaccine-preventable viruses.
Oh, this category includes pathogens like measles and polio.
Yes. And ideally, in a highly vaccinated population, the wastewater signal for these viruses should be absolute zero.
Complete silence on the dashboard. So if a signal for measles appears, it's a glaring failure of the public health shield.
It is an immediate crisis. If TextWeb detects a sudden, sustained spike of measles RNA in a specific city sector, it is a flashing red light indicating a collapse of herd immunity.
It tells officials that vaccination rates in that specific localized area have somehow dropped below the critical threshold.
Right. It allows public health teams to aggressively deploy modal vaccination clinics and educational campaigns to that exact neighborhood to extinguish the spark before it becomes a raging clinical fire.
That is incredibly precise. But then we cross the threshold into Category 4, the catastrophic threats.
This is where the protocols become extremely rigid and immediate. Ebola, West Nile virus, dengue fever, highly pathogenic avian influenza.
Scary ones.
These are pathogens with devastating mortality rates that are not endemically sustained in the typical U.S. population. If a signal for a Category 4 virus appears on the dashboard, there is no waiting around to see a multi-day trend.
You act immediately.
It triggers immediate laboratory validation protocols. They run the tests again. If confirmed, the public health network is instantly notified so that frontline clinicians are put on high alert to look for weird symptoms they would normally never expect to see.
Preventing crucial misdiagnoses in emergency rooms. If someone comes in with a fever and the doctor knows Ebola is in the water, they treat it very differently.
Exactly. And finally, sitting at the very top of the matrix is Category 5.
Disease X.
The detection of a completely novel viral signature or a heavily mutated variant of a known virus that indicates it may evade current immunity. This is where the noise-canceling power of the WVDB we discussed is deployed.
Because you wouldn't even see Category 5 without the database.
Right. Detecting a Category 5 threat would trigger national and international emergency protocols to isolate and sequence the threat before a single patient succumbs to it.
It is a brilliantly logical system. It translates this incomprehensible stream of ATCG genetic letters into a clear, color-coded threat matrix.
It's a great model.
But as we dig deeper into our sources, we uncover a rather depressing reality about the current state of WBE.
The adoption rate.
Right. Despite this incredibly powerful early warning system existing, there is a massive chasm in communication. A recent survey conducted jointly by the CDC and the Infectious Diseases Society of America revealed a really startling statistic. Only 22% of infectious disease doctors actively review wastewater data in their practice.
Only 22%. That means nearly 80% of the highly specialized doctors fighting on the front lines of disease are just ignoring the most advanced surveillance tool in human history.
Why is that happening? I mean, if I am an infectious disease specialist and my entire job is to save patients from outbreaks, wouldn't I desperately want to know what is coming down the pipeline next week?
They absolutely want to know. The disconnect is not driven by a lack of interest. It is driven by a lack of interpretability.
Okay, unpack that for me.
Well, imagine you are an exhausted, overworked clinician in an emergency room. You pull up a state health dashboard on your phone and it tells you, SARS-CoV-2 viral load in the Northside treatment plant is currently at 50,000 copies per liter.
What does that mean to a doctor? I mean, does 50,000 copies mean 3 people in the neighborhood have a mild cough? Or does it mean 3,000 people are about to flood the emergency room with severe pneumonia?
That is the exact problem. To a clinician, a raw genetic copy count is just an abstract, useless number. It does not translate into how many ventilators do I need to prep for Tuesday morning?
Right.
The field currently lacks standardized, easily translatable thresholds. Scientists are furiously working to solve this by developing fiercely complex mathematical models known as reverse QMRA.
Reverse QMRA. Okay, we definitely need to unpack the math behind this because it sounds like the holy grail for making this data useful to the medical community.
It is. QMRA stands for Quantitative Microbial Risk Assessment. Traditionally, engineers use it to calculate forward-looking risks.
Forward-looking. Give me an example.
For example, if a water treatment plant detects a specific concentration of a pathogen in the clean drinking water supply, QMRA math calculates the exact statistical probability that a person drinking an 8-ounce glass of that water will contract an infection.
Oh, I see. It calculates the risk to the individual based on what's in the environment.
Yes, but reverse QMRA attempts to run that incredibly complex mathematics backward.
Backward. So if we detect a specific viral load, say that 50,000 copies per liter we mentioned, in the massive aggregate volume of a municipal sewer, how many actively infected human beings must exist in that population to generate that exact genetic signal?
Exactly. You were trying to guess the number of sick people based on the amount of virus in the pipe.
But that math seems almost impossible to standardize given all the variables in a city.
It is a staggering mathematical challenge. It gives statisticians nightmares. To build an accurate reverse QMRA model, they have to account for a dizzying array of dynamic variables. First, you have human shedding variance.
Setting variance.
A person infected with COVID-19 might shed billions of viral copies in their feces, while another infected person sheds only millions.
So the biological input isn't even standard.
Right. And some viruses are shed primarily in urine, others in stool, others through respiratory mucus that gets washed down the sink when you brush your teeth.
Okay, so the human side is highly variable. What about the journey through the pipes?
The sewer kinetics are just as complex. You have to factor in the flow velocity of the water, the diameter of the pipes, and the total residence time.
Residence time.
How long it takes the waste to travel from a toilet in the suburbs all the way to the central treatment plant. If it rained heavily yesterday, stormwater infiltration massively dilutes the sample, artificially lowering the viral count per liter.
Because it's mixed with rainwater. And earlier you mentioned that RNA degrades in the sewer, so they have to mathematically model the destruction of the evidence while it travels.
Exactly. They have to calculate the decay kinetics. This involves factoring in the ambient temperature of the wastewater, the presence of industrial chemical discharges that might destroy the RNA, the degradation caused by UV light if the channels are open to the sky, and even the shearing forces of the water flow itself that physically tear the fragile RNA strands apart.
Wow. So 50,000 copies per liter detected in the middle of a hot summer might mean 10 people are sick because the heat destroyed most of the RNA. But that exact same 50,000 copies detected in the dead of winter when the cold preserves the RNA better might mean 100 people are sick.
Precisely. The math changes every single day based on the weather and the pipe conditions until the reverse QMRA models are perfected and can reliably translate raw viral copy numbers into accurate, estimated clinical case counts. The data remains too abstract for a busy local doctor to act upon confidently.
Which makes total sense.
Which is why initiatives like TextWeb are dedicating immense resources not just to the sequencing chemistry in the lab, but to user interface and user experience on the computer side.
Right, building dashboards.
They are building highly intuitive, graphically clear dashboards. They are trying to bridge the translation gap between the bioinformaticians who extract the genetic data and the clinicians who need to know how many beds to prep.
Because the science is only as valuable as our ability to comprehend and act upon it.
Yeah.
But, this profound capability to extract data from our collective waste forces us to confront an issue we really cannot ignore.
An ethical minefield.
Yes. Whenever humanity builds a surveillance apparatus, this powerful system technically capable of reading the intimate biological output of an entire civilization, we inevitably step into a regulatory and ethical minefield. The technology has, as usual, completely outpaced the law.
This raises what is perhaps the most profound question of this entire endeavor. Just because we possess the technological capability to sequence and analyze absolutely everything we flush, does that mean we should?
It's a great question. We have spent this deep dive celebrating WBE because, fundamentally, it is inherently anonymous. As you said, it is aggregate data. You are just one tiny drop in an ocean of 1 million people at the treatment plant. Nobody knows it's you.
Right. At the treatment plant level, it's very safe, ethically.
But our sources analyzing the ethical frameworks warn about what happens when the microscope gets tighter, what happens when the ocean becomes a puddle.
The ethical peril scales inversely with the size of the catchment area.
What does that mean in practice?
Well, it is universally accepted as ethical to sample the main influent of a treatment plant serving a major city like New York or Chicago.
Yeah.
The data is entirely de-identified. But the sequencing technology is highly portable and incredibly precise.
But you don't have to stay at the main plant.
No. What happens when you move the sampling equipment upstream? What if you sampled the specific sewer line exiting a single university dormitory housing only 50 students? What if you sampled the wastewater holding tank of a single commercial airliner when it lands? What if you sample the outflow of a specific corporate office building?
Right. Because if you sample a dorm of 50 students and detect a high signal for illicit narcotics, or maybe a heavily stigmatized disease, you haven't technically identified a specific individual by name, but you have drawn a very tight, highly suspicious circle around a very small group of people.
And that exact scenario leads directly into the primary ethical fear, highlighted by legal scholars looking at WBE, function creep.
Function creep, which is the concept that a powerful tool built and justified for one noble purpose slowly, inevitably gets utilized for other more invasive purposes.
Exactly. Currently, community-level wastewater surveillance is largely unregulated because human waste deposited into a municipal sewer is legally considered abandoned property.
So no one owns it. It does not require informed consent to collect and analyze.
Right. And it was built to protect public health. But consider the potential for function creep. What happens when local law enforcement agencies decide they want to use untargeted sequencing to hunt for the chemical precursors of illicit drug manufacturing in specific low-income neighborhoods?
It sounds like a massive overreach.
It gets worse. What happens when a massive corporation decides to monitor the aggregate health, stress hormones, or dietary habits of their employees by tapping the main sewer line of their corporate campus?
We noticed our employees are eating too much junk food based on the sewer data. That's terrifying.
What are the implications for international espionage if intelligence agencies discreetly monitor the wastewater of foreign embassies to track the health of diplomats?
Using biological waste as an unregulated tool for a surveillance state. If the public ever perceives that their own biology is being weaponized against them in that way, the public trust will evaporate instantly.
Instantly.
And it will destroy the legitimate, life-saving public health applications of the entire system. People will actively sabotage it.
The loss of public trust is the absolute greatest threat to WBE right now, and our sources point out that this threat is compounded by the danger of stigmatization.
How so?
It is one thing to publicly track the rise of influenza or RSV. Everyone gets the flu. But as the sequencing resolution improves, public health departments are increasingly interested in tracking sexually transmitted infections, such as HIV or Mpox.
Which requires extreme sensitivity.
Absolute discretion.
Because if a city health dashboard publicly announces that a specific zip code, perhaps a neighborhood with a high concentration of marginalized groups, is currently experiencing a massive surge in Mpox based on wastewater data, the resulting media frenzy and public reaction could lead to severe economic and social discrimination against the people living there.
The ethical guidelines surrounding WBE explicitly emphasize this, engaging with local community leaders and vulnerable populations before tracking and publishing data on stigmatized diseases is absolutely paramount. Transparency and context are the only shields against stigma.
And even when public health officials have the best of intentions and follow all the ethical guidelines perfectly, a fundamental lack of scientific literacy in the general public creates a vacuum that is very quickly filled by misinformation.
It does. It's a huge problem.
Yeah.
And to understand how easily this communication breaks down, we must examine a highly illustrative incident from December of 2023.
Right, the Breitbart example. And I want to be clear here. We are looking at this strictly through the lens of science communication. It perfectly highlights the perilous gap between complex laboratory reality and public interpretation.
Let's examine the mechanics of what occurred impartially.
Go ahead.
In December 2023, the news organization Breitbart published a report stating that high levels of COVID-19 had been detected in the nation's water supply.
Okay, let's pause there. From a strictly biological perspective, based on everything we've discussed today about WBE, they were referring to the detection of fragmented, deactivated viral RNA in untreated municipal wastewater, the raw sewage.
Correct. That was the underlying scientific data being referenced. However, the phrase water supply is a broadly interpreted term.
Very broad.
To a bioengineer or a city planner, water supply might encompass the entire hydrologic cycle of a city, including the sewer output.
But to the average layperson reading a headline, Water supply almost exclusively evokes the clean, treated drinking water coming out of their kitchen tap.
Exactly, which led to a massive immediate breakdown in public understanding. The reporting sparked rapid, rampant speculation across social media platforms.
People began theorizing that the government or nefarious actors were intentionally infecting the public drinking water infrastructure with the live COVID-19 virus.
Which is, we have to state, biologically and infrastructurally impossible.
Why is it impossible?
Because a fragile respiratory virus like SARS-CoV-2 simply cannot survive the rigorous chemical chlorination and UV filtration of a modern drinking water treatment plant. It's obliterated.
Furthermore, the public completely misunderstood what wastewater surveillance actually measures in the first place.
Yes, WBE does not detect infectious live viruses floating in the water. It detects the shattered, deactivated genetic fragments, the RNA shreds we talked about, left over after the virus has already been destroyed by the harsh sewer environment.
You can't catch COVID from an RNA fragment.
No, it's just a chemical ghost. But the incident proves that the science does not exist in a vacuum. You can build the most advanced supercomputer database in the world. You can run perfect de novo assemblies. But if the public doesn't understand the difference between raw sewage and treated tap water, or the difference between a harmless RNA fragment and a live infectious pathogen, fear will override the data.
Every single time.
It is a textbook lesson for public health agencies. If WBE is going to succeed and expand as a permanent fixture of global health security, the scientific community must become proactive educators.
They can't just publish the data. They have to explain it.
They must mandate precise language in their reporting, design highly transparent dashboards, and constantly explain the mechanisms of the science to the public. If they fail to bridge that communication gap, misinformation and fear will inevitably derail the entire effort.
It is a sobering reminder that deploying a new technology is often much, much harder than actually inventing it. Well, we have covered an absolute marathon of information today.
We covered a lot of ground, yes.
Let's pull the threads of this incredible journey together. We began by tracing the historical roots of this science, moving past John Snow and deep into the 1940s, where scientists laboriously dragged those Morse swabs through the sewers to track the hidden spread of polio.
The origin of it all.
We saw how the COVID-19 pandemic catalyzed that slow manual process into a global, real-time intelligence network capable of completely bypassing the inherent biases and failures of clinical testing.
We followed the CRISPR consortium into the dark matter. We explored the severe limitations of targeted barcode scanners and the read-based wall that left 56% of our genetic waste an unreadable mystery.
We walked through the staggering computational brute force required to perform de novo assembly on hundreds of billions of genetic fragments, relying on complex math like debris graphs to stitch together overlapping shreds of an alien language.
We witnessed the agonizing scientific rigor required to purge chimeric Frankenstein viruses, resulting in the painful deletion of half their data. But out of that sacrifice emerged the wastewater virome database.
The WVDB. A catalog of over 21,000 near-complete viral genomes, including 17,000 completely novel biological entities that science had literally never seen before.
We discovered a hidden, chaotic ecosystem down there, dominated by bacteria-hunting phages and remarkably resilient plant viruses passing through our diets completely unharmed.
And crucially, we explored how that database acts as a digital noise-canceling filter, allowing bioinformaticians to digitally subtract the benign background static so that the faint, terrifying signal of disease X can actually be heard clearly.
We saw how networks like TextWeb are translating that complex data into a 5-tier triage matrix, while wrestling with the fiercely complex mathematics of reverse QMRA to make the viral counts actionable for frontline doctors.
And finally, we confronted the ethical tightrope. The pressing need to guard against function creep in small catchment areas, the responsibility to protect vulnerable communities from data-driven stigma, and the absolute necessity of clear science communication to prevent the kind of public panic and misinformation we saw when complex data is misunderstood.
It really has been a staggering, paradigm-shifting journey to discuss.
It has, but before we sign off, I know you have one final, highly provocative thought for us to mull over. Something that builds on the data we've discussed today, but points the compass in a completely different, incredibly hopeful direction.
I do. I want to return to what I think is the most surprising statistical finding in the entire WVDB paper.
Okay, what is it?
The revelation that 79% of the RNA viruses discovered in our wastewater are bacteriophages. Viruses that have evolved over billions of years for the sole, highly specific purpose of actively hunting, attacking, and killing bacteria.
Right, the microscopic predators.
Now, connect that massive viral army we just discovered to a broader global picture. One of the absolute greatest looming crises in modern healthcare right now is antimicrobial resistance, or AMR.
The rise of the superbugs. Bacterial strains that have mutated and evolved to defeat every single antibiotic drug we have in our medical arsenal.
Exactly. We are rapidly approaching a terrifying post-antibiotic era where simple infections, like a scraped knee or routine surgery, could once again become lethal.
Because the drugs just don't work anymore.
But right beneath our feet, flowing endlessly through our municipal sewers, is a vast, largely untapped biological armory.
Oh, wow.
The CASPR researchers just cataloged 16,000 newly discovered phages that are absolute masters of bacterial destruction. This raises a breathtaking possibility for science.
I see where you're going with this.
As we map this dark matter, are we merely building a passive surveillance tool to watch for disease? Or, by cataloging the staggering diversity of predatory phages, are we actually looking at the raw blueprints for the next generation of medicines?
Could the ultimate cure for the superbug crisis, the next evolution of highly targeted phage therapy, be waiting down there?
Exactly, waiting to be engineered out of the very viruses we are currently flushing away without a second thought.
What an incredible paradigm shift. We aren't just looking into the sewers to find the next great threat. We might be actively mapping the next great cure.
The solutions might literally be in our waste.
That is amazing. Thank you so much for joining us on this deep dive. And to our listeners, the next time you walk into a room, take care of business, push a handle, and watch the water swirl down the drain. I really hope you remember the staggering complexity of what happens next.
It is a whole other world down there.
It's not the end of the line at all. It is just the beginning of a vast, invisible story. Keep questioning the unseen world around you. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.